Hello everyone, and welcome to our lecture on microscopes. Now note that the microscopes lab is lab number 3. We are skipping lab number 2, the topic of which is experimentation. You're welcome to read it on your own, but there will not be a quiz on it, and you are not responsible for knowing the contents of lab 2. All right, so let's talk about microscopes. If you've read through the lab or this week's announcement, you've probably noticed that for exercise number 1, you're supposed to watch a video. Now that video is linked in this week's announcement. It takes you to a YouTube video from our very own Penguin prof, which is Professor Valerie Pennington. She works at Southwestern. Make sure you watch that very first video, the first video in this week's announcement. You watch it before watching this lecture, all right? I'm going to go through the contents of this lecture under the assumption that you have already watched the Penguin Prof's video on microscopy. Now, when you watch that video, she's going to take you through a number of topics that correspond nicely with the lab manual itself. She's going to talk you through the different types of microscopes. Then she's going to talk you through the various microscope parts. She's going to talk you through how to use a microscope, what your best practices are, meaning if we were in the lab, what would be the correct way, the most appropriate way to handle the microscope as a tool. And then the latter half of the video talks about how to find stuff using a microscope. And it goes through various technical terms you need to understand in order to fully grasp how microscopes do their job. She talks you through things like resolution, magnification, field of view, depth of field, and light intensity. Now, these are the topics that I think are the most important takeaways from this lab. So the rest of this lecture is going to focus here on topic number 4. Now, when you go about using a microscope, there are a few key parts you have to pay attention to in order to understand exactly how much an image is being magnified. Those parts of the microscope are referred to as the lenses. There's 2 different types of lens. First, there are the ocular lenses, which are right next to your eye. So this is the ocular lens on this particular microscope, and it's the one that goes right next to your eyeball. So this is what you would actually be looking through if you were using this microscope. Ocular means eye. But the next type of lens that's important is the one that's next to the object that you're looking at. So whatever's down here on our glass slide that we're trying to magnify, that's our object. The lenses that are closest to that object are referred to as objective lenses. Okay, so these 3 lenses here in blue are your objective lenses. Now notice the ocular lenses and the objective lenses have numbers written on them. This is the amount of magnification you get from each of these lenses. Ocular lenses are usually either 5x or 10x. All of ours in our examples are going to be 10x. This means that they magnify an image 10 times. They make it appear 10 times larger than it is in real life. Objective lenses have a wide range of different magnifications. The ones in the lab, in our lab, that you would have encountered would have been 4x for the smallest, 10x for the medium, and 40x for the largest. Okay, so the smallest of these will magnify an image by itself by 4 times. The next would magnify an image by 10 times, and the largest would magnify an image by 40 times. Now notice, when you look through this microscope, you are looking through first the ocular lens and then one of the objective lenses. That means both of those lenses are going to be magnifying the image that you're seeing. The next thing you need to understand how to do is to calculate what's called your total magnification. Your total magnification is going to be a combination of the effects of your ocular and objective lenses. Now, how do you deal with these numbers? Do you add them together to get your total magnification? Do you divide them, multiply them? The answer is you multiply. To determine what the total magnification is of the image you're seeing, you multiply the value of your ocular lens by the value of your objective lens. So if your ocular lens is 10x, for example, and you're looking through your 4x objective lens, that means your total magnification for the image that your eye is seeing is going to be 40x. Now, what if you're using that medium power objective lens, that 10x lens? Well, in that case, your ocular lens will give you 10x magnification, 10 times. Your objective lens is also giving you an additional 10x of magnification. Multiply the two together and you get a total of 100x. We can do the last one as well. Again, your ocular lens is going to be 10x. Here's a big hint. Your ocular lens will always be 10x for the purposes of our class. Let's say you're using your largest objective lens, 40x. Multiply the two together and you'll see that this particular microscope has a maximum magnification of 400x. That means if you're using its most powerful lenses, the microscope depicted here can magnify things 400 times. And the purpose of microscopes is to do just that. It's to increase the visual size of tiny objects to let you better examine them. But there are some interesting side effects of this process of magnification. There are other things that change in the image you're seeing as it's being magnified. Now, I'm going to show you a few examples of these things using some diagrams of actual slides. So imagine that on the left side here, you are seeing a glass slide that you've placed a water droplet on. And perhaps that water droplet came out of a pond. And you know there are microorganisms, tiny living things, floating around in this water droplet. Okay? And you're interested in seeing them up close. We'll do those three. Okay. Now, from above, it looks like all of these little critters, these little living things in your water droplet, are all basically on the same level. They're an equal distance away from your eye. But if we were to look at this slide from a side view, if we were able to look very, very closely at the slide from the side, what you would see is that the water droplet isn't flat. It isn't just plastered down flat on top of that piece of glass. Instead, the water droplet is a three-dimensional space sitting on top of that glass slide. Okay? And floating in that water droplet, you have your microorganisms. But one of them is a lot closer to the top of the droplet than the others. One of them is a lot closer to the bottom, sort of sticking to the glass. And our last one is sort of halfway in between, kind of in the middle of the droplet. Always remember that whatever you have on your slide that you're trying to examine, it's a three-dimensional thing. Nothing is really two-dimensional when you're looking at it under a microscope. You have to keep in mind that your sample here not only has width to it, okay, so it doesn't just cover a flat space like this, it also has depth. it has a vertical height component to it as well. Right? And this is what's going to create some interesting effects when we try to magnify the images, or I'm sorry, when we try to magnify the organisms on this slide. Right? All right, first thing I want to show you is something called field of view. Your field of view is the area of the slide you can actually see through the eyepiece of the microscope. So if I were to redraw our slide here with our water droplet and our little organisms, here's what our field of view would look like. If we were using our lowest magnification, which would be our 4x lens, and we looked through the eyepiece of our microscope, what we would see through that circular eyepiece is a circular image that includes about that much of the slide. That's how much of the slide we could actually see if we were using the 4x objective lens. But let's say we wanted to get closer. We wanted to have a more magnified view of this slide. Well, in that case, we could rotate our nose piece and change our objective lens to our 10x. That would zoom in, it would magnify the image, so we'd get a much closer look, say, at this little yellow guy using the 10x objective lens. But notice we're seeing a lot less of the total slide overall. We are only now seeing this area inside this innermost gray circle, right? That's what happens when you increase magnification. You zoom in on a certain area of the slide, but it means you see less of the rest of the slide overall. Now we could go one level further. We could zoom in on just this part of the slide using our 40x objective lens. Right? That's our most powerful objective lens. This gives us our highest magnification. My tablet's not happy about it. It can't even zoom in that far. But we would just be seeing what's inside of this innermost gray circle using that objective lens. It's a nice magnified view of that little yellow organism, but it completely excludes everything else on the slide. So you can see how your field of view, the total amount of the slide you see, gets smaller and smaller the higher your magnification. Okay. This is the first relationship between different factors that affect microscopy that you need to know. Okay. The relationship is that as magnification increases, your field of view, FOV, is going to decrease. So the more magnified your image, the more you zoom in, the smaller your field of view, which makes sense. If you're magnifying something, you're zooming in on a small area of it, right? Now, for this reason, when you are first starting to search a slide, looking for things on it, you always want to start your search using your 4x objective lens. Okay, this is sometimes called the scanning lens for this reason. The reason you want to search using this lens is because it shows you the largest area of the slide. If you tried to start your search using that tiny little 40x lens, you'd have to move it back and forth over and over again, hoping to encounter something on the slide. It's too small of a lens to effectively use as a searching tool. You want to have a big lens, a big wide field of view when you're first looking for the specimens on your slide. All right, so whenever you start searching for things using a microscope, start with the lowest power objective lens, your scanning lens. All right. Here's the next factor that affects your magnification and your use of magnification while using a microscope. Okay, so that was number 1. This is number 2. This is called depth of field. Your depth of field is how much of the vertical area on the slide is in focus. Now here's what's interesting about your depth of field. Just like field of view, as your magnification increases, your depth of field is going to decrease. Same relationship as field of view. So let's scan back up to our original diagram of our slides here. Now, here's what I want you to see. Notice on the diagram on the right, again, that we have vertical locations on our droplet of water. So we don't just have objects that are further to the left or further to the right. We also have objects or organisms that are located higher up or lower down. This is the vertical location of our specimens, and this is what is affected by depth of field. At lower magnifications, our entire depth of field, or close to it, is in focus. But our field of view is also very large, so nothing's all that magnified. Okay? So at low magnification, here's what we might be seeing through our lens. Okay? Everything will be probably nice and crisp and in focus, but everything will also be very small. Okay? So at this low magnification, our depth of field is great. Everything is in focus. Things that are shallow, things that are deep, everything's in focus at low magnification. And our field of view is very big. So we're seeing a large proportion of the slide. At this level of magnification, everything between these two yellow lines is in focus, right? And look what happens if we go to high magnification. Our field of view is going to be much smaller. It says magnification increases, field of view decreases. Let's focus on the red guy. Let's say we have adjusted our focus, so we're looking at that red microorganism. At high magnification, we're going to be much closer to it. We're going to see it in more detail. But here's the trick. Now, our depth of field, which is what both of these brackets are representing. Both of these represent our depth of field. Our depth of field is going to be much smaller. Now, only the section of the water droplet between these two red lines is going to be in focus. That's it. If we were to move our lens, so this is our lens up here. We're currently looking down here at this red guy. If we were to slide our lens over so that it was directly above this pink organism, what we would see is a very fuzzy, out of focus image. And the reason is this pink guy is above the depth of field that's actually in focus for us. Okay? Now, in order for us to see the pink guy clearly, we are literally going to have to move the slide up towards this lens. We're going to have to bring that the field of view the depth of field, this area that's in focus, we're going to have to bring it up so that the pink guy is within it in order for him to be in focus. That depth of field is much, much smaller at higher magnifications. It means there's a much narrower slice of the image that's actually in focus when you're at high magnification. Okay? It's another one of these inverse relationships between magnification and another characteristic of the image. So far we have seen that field of view, the area of the slide you can actually see, is inversely related to magnification. You see now too that depth of field, how much of the vertical area on the slide is in focus, that's also inversely related to magnification. And here's our last characteristic, light intensity. So light intensity is literally how brightly lit is your image. Okay. So take a look at this diagram. Once again, this little green rectangle represents your slide as seen from the side. It's a lateral view. These are your objective lenses up here. Now notice our 4x lens, our lowest magnification lens, has a big gap between it and the slide itself. There's this big area of empty space. Now this large gap allows a whole lot of light to shine in on the surface of the slide. And that light is going to illuminate, literally, whatever it is that you're looking at here on your slide. The 4x lens, because it's so far away from the slide, allows a lot of light to shine on it. Now look over here for contrast at the 40x objective lens, our highest magnification lens. Look how big the gap is here between the lens and the slide. It's tiny. Almost no light is going to be able to penetrate here to shine on your slide. The 40x lens literally casts a shadow on the slide, making it very, very dark and hard to see. Because of this, most microscopes come with some sort of light source, like what you see drawn in here, that illuminates the image from below, okay, helping to compensate for some of that shadowing effect. But what this means is that as you increase your magnification, you have to manually brighten up your image using this light source. Otherwise, it'll be too dark to see. All right, those are the major ideas that I wanted to go over in lecture this week. Now, just to review, there are other things that you're responsible for knowing for the quiz that I didn't go over in this lecture. Things like the different types of microscopes, the different parts of the microscope plus their functions. You can find these on pages 49 and 50 in the lab manual. The functions of the different parts, the jobs they do, is found on page 50. How to use a microscope, meaning the best practices for how to actually use the different knobs, use the different adjustments. And then finally, how to find stuff using a microscope. Now, while we went over the majority of things in this section, some of the more easy to understand concepts like resolution I didn't talk about, but they are explained pretty well in the lab manual itself. Now, also keep in mind that there are additional videos in this week's announcement that you'll need to use in order to complete some of the sections of the lab. The announcement talks you through which video links you will want to use for which lab exercises. And also keep in mind that there are certain parts of the lab that you're going to be skipping. So make sure you go to our Canvas page, go to Assignments, and click on the assignment for this lab, which is lab number 3, and look at the pages you have to upload and the pages to skip. Make sure you have those written down somewhere while you're working through the lab so you don't complete something that you aren't responsible for, or you don't skip something that's necessary. All right, that's it, and I'll see you in the lecture for lab number 4.